My name is Jenny Pierson. I'm a Senior Ecologist with Australian Wildlife Conservancy. And as the name conveys, I am actually not a botanist. I'm on AWC's National Science Team, and I've worked closely. We do have two botanists on the science team, but they live up by Cairns, and so I've worked really closely with them to be able to tell you a little bit about the work that we're doing, particularly inside and outside our predator-proof fences. I'm going to be using some notes today to get the plant details right, because I know that's probably what you guys are interested in, and that's, like I said, not my specialty. So basically, Australian Wildlife Conservancy is one of the essentially largest conservation organizations in Australia. Our mission is to conserve Australian wildlife and its habitat. And so with that, we really recognize that its habitat is plants as well. And in the last few years, we've really broadened out to, we have this really large-scale eco-health monitoring program where on all of our sites we monitor the health of the biodiversity, and we have started targeting threatened species indicators, including plants, as well as the vegetation structure there. And so it is in our purview now to make sure that we are monitoring the habitat and making sure that we're taking care of biodiversity writ large. At this stage, Australian Wildlife Conservancy manages, through partnerships or owning sanctuaries, close to 13 million hectares. So it's incredibly large scale what we're doing, and that's really what we aspire is to make conservation differences, what we call it, at scale. Today what I want to talk to you about is one of the areas that we specialize in, and that's the reintroduction of mammals that have gone extinct over time locally. And a lot of these animals, particularly the ones that we target for reintroductions, are what we call this critical weight range mammal that's been targeted by feral foxes and cats. And that's because we can actually ameliorate this threat. We can build these large predator-proof fences, and we can take all the feral animals out of there, and then the threat's gone. And that's one of the big challenges with reintroduction, is it's hard to do a reintroduction to recover the species if you can't get rid of the threat. So it's a really powerful tool we have to actually make sure we can recover these species. Now, AWC, these predator-proof fences are becoming more and more common. AWC is not the only one who does them, but we probably have more than anybody else, and we're starting to do them at a scale that's incredibly large. So the fences that we have are anywhere from 250 hectares up to 10,000 hectares, and most of ours are over 1,000 hectares. So again, we're able to do this where we can actually have functioning systems and large populations of mammals. And this makes it so we actually have sources for other sites. We really are trying to turn the tide for extinctions for these species. Now, because we're making these essentially mainland islands, these fenced islands, one of the things is even though they're at scale, the populations are going to grow and come down. And so what we wanted to do was start to understand what's happening to the habitat in general, what those changes are. Now, what we don't know is kind of what the pre-European history was. And so we're able to look at inside and outside the fences of these areas. And outside the fences is still in AWC reserves. So we manage these large plots of land and we put the fences on them. But for instance, at New Haven, our site in Central Australia, our fenced area is close to 10,000 hectares. New Haven itself is 100,000 hectares. So we're able to look inside and outside the fence, but still on conservation reserve land. And so that means we can really look at some of the targeted differences for the reintroduced species. And then outside the fence, some of the things that are different tend to be, perhaps we haven't been able to fully eradicate some of the feral herbivores, where we are able to do that inside the fence. So things like the pigs and goats, although we manage them at those scales, sometimes we're not able to fully eradicate them. So differences in terms of what you're looking at there. So we've gone in and we've put plots at all of our sites that we have fences. We've instigated inside and outside the fence, paired long-term monitoring plots to start looking at these vegetation changes. Today, I want to talk to you about three different things essentially. I'm going to tell you some results from our two longest-term sites that we have the fences, and that's Yucamura in South Australia and Scotia in southwest New South Wales. And then I'm going to focus in on some more particular results that we have from one of our species. And then finally, I'm going to tell you about some work we're doing in the Pilliga, which I think some of you may have gone to some field trips there and maybe going to some field trips there still. And that's where we've had a chance to start doing some targeted monitoring before the species go in. So Yucamura and Scotia were both fence reserves before AWC took over the management. So the fence at Yucamura is probably one of the oldest ones in Australia. It was built in 1989, and there were some captive breeding programs that happened there. In the early 2000s, AWC took over and were able to shore up the fences, release the animals from the captive breeding sites, and there's been reintroduced some of these digging mammals there. Now a lot of these mammals that get reintroduced, these critical weight range mammals, tend to be the digging mammals that turn over huge amounts of soil. So there's been bandicoots, bilbies, bettongs. The different studies show up to 1,000 kilograms of soil per year per individual. It's between the diggings and the burrows that they're making. So incredible. When we call them ecosystem engineers, it's a really genuine term in terms of they're really having effects. So some of the things that we're seeing, like I said, we can't really say, pre-European what our target is, because before these animals went in, we have differences in terms of the feral herbivores compacting that soil, those hoofed animals on the landscape. We have rabbits that have exploded and completely degraded the landscape, and digging in very different ways in terms of the soil turnover. So we don't necessarily have a target of what's good or bad or pre-European not. What we're trying to figure out is kind of just what's, quantify what we're seeing in terms of what's happening. So what we did, so I'll read some notes now to make sure to give you some good details, is try to look at the sites of the different habitat types. So basically, Yucamura has two widespread vegetation types. It's got the mallee woodland, which is the brown loam over calcrete with the Eucalyptus gracilis, oleosa, and brachycalyx, and that's over Muller's daisy bush, blue bush daisy, and the chenopod's. And then it's got the myoporum shrubland, and that's basically scattered myoporum with cassia, stiff westringia, and then the narrow-leaf hawkbush and different grasses. Scotia has three widespread vegetation types. It's got the mallee triodia, and that's basically the Eucalyptus socialis, dumosa, incrusata, and that's over spinifex. And then it's got the mallee shrub, which is dominated by the Eucalyptus oleosa with cassia, emu bush, and the chenopod's. And finally, you have the Bala woodlands there, which is the casuarina papa with chenopod's and melissa daisy bush. So basically, we had 28 plots at Yucamura, 16 in the mallee, and 12 in the myoporum, and we split those paired inside, outside, long-term vegetation monitoring plots. Scotia, similarly, we had 32 plots spread equally among the three vegetation types, paired for each of those sites inside and outside. And then in terms of our long-term monitoring, we did 20 by 100 meter plots with transects every 5 meters. When we looked at species composition, we actually recorded the species, every single species we did, with their frequency, in 121 by 1 meter plots. And then we used a laser to record canopy intercepts to look at structure. I recorded the different type, is it bare ground, is it shrub, is it grass, all that kind of stuff. So at Scotia, we recorded a total of 70 species, but 84% of those were at a frequency less than 2.5%, so we weren't able to include those in the analysis. Similarly, at Yucamura, we had 106 species, with 71% of those were less than 2.5%. So we really had to focus on the more common species to be able to start teasing about some of these differences. So this is what we found over here. Hopefully you guys can see. This is a poster showing some of the results from Scotia. And these are just box plots showing the median and then the 95% spread in terms of what we found, and then the tails on the end. And we've got two different treatments at Scotia, and that's because we actually have two fenced areas. The original fence that was there, we call it in 2004 because that's when that one was finished in terms of us shoring it up. We decided to double the area at Scotia, but we kept those fences separate because that actually allows us to do different things in the different sides, and that's called in 2008. Now the really key thing about these differences is the different species that's in them. So between both sites, we've got numbats, we have bilbies, we have brush-tailed bettongs, but burrowing bettongs or booties are only at Yucamura and also in this in 2004. It's not in 2008, so the one that was finished in 2008. The other difference is Scotia has bridle nail-tailed wallabies and Yucamura has brush-tailed possums that were reintroduced, and those become very common and dense when they're inside fences, no matter where you are. So here again, what you can see is basically, generally for species richness, we saw higher outside than inside the fence when you look at in that 2004. But when you look inside the 2008 in the mallee areas, you actually don't see a difference. So again, if you remember, that main difference there is going to be the timing of it, 2008, but that's still a long time. But also those burrowing bettongs and booties that become quite common. At Yucamura, what we can see is that we did see a difference both in species richness, but also in functional groups. We didn't see a difference in the functional groups in Scotia inside and outside the fence between either area, but we did see a lower frequency of the chenopods and also some of the low shrubs at Yucamura. But the other thing that we saw was that the introduced geophytes, the thread iris, were also less frequent inside Yucamura. So luckily, the animals in there were eating everything, so that was helpful. And so these are the differences in terms of the two more common ones that we could look at. We found that this ruby saltbush and the pink-seeded bluebush were both less frequent inside the fence than outside the fence. Now this top one, the ruby saltbush, that's something that again, other places with these reintroduced animals, we know that's a forage food, so not terribly surprising that this yummy plant was being eaten. This one hadn't been shown before as being a common forage plant, but again, we're surmising because of its similarity in terms of that fleshy leaves that it is probably being foraged by these animals, and that's the difference in the frequency that we're seeing. Generally saw a bit more bare ground, not surprising, and fewer woody seedlings. Now what's interesting is looking at some of these differences in terms of why we saw Scotia and Yucamura maybe different. Yucamura, again, is a bit smaller, and it's, again, probably one of the oldest fence sites in Australia. So, before AWC had it for a long time, it was Earth Sanctuaries Limited. And so it's had different management since at least 1989. And it had a very dense number of captive breeding animals in there as well in that previous time. But the other thing that's really interesting is this pattern we see with the burrowing bettongs or booties. Now, booties are a species that can become very common inside fences or on islands. We have them on Foria Island, and there's about 15 to 20,000. And if you go back and read some of the early naturalist descriptions, booties are one of the ones in WA that are described as just being hundreds. They get inside your shed. They get people had them as pets. They were just so common everywhere. So whether or not this is a false economy having an offense, or if it's just when booties are around, the boom for booties is a really big boom, and they're just an incredibly common species. So I wanted to tell you a little bit about some research that one of the AWC ecologists, Bryony Palmer, did her PhD on, and that was looking at the effects. So she really zeroed in on particularly warrens of booties, and she looked at this at Foria Island, where we have them, at Yucamura, and also at Manawa, one of the sites in Western Australia that UBCA manages. We've had particular questions. I haven't printed out her particular results. I'll do my best. And what's interesting is she found that there was actually, this incredible nutrient cycling effect that she saw. There was higher nutrients in the tailings of those warrens, and so in the warrens you see the entrance, and then you have this huge tailing when they dig it up. But also higher soil moisture. And in these dry arid lands, that's an incredible thing to have the higher soil moisture. We saw higher vegetation cover with all of that, but it wasn't favored by native. So she saw both non-native and native vegetation was equally benefiting from these nutrients and that soil moisture. But you did have more grasses, all that kind of stuff, particularly at Yucamura. And so it is really interesting in terms of, again, like I said, we don't have this target of what we're going for in terms of pre-European, not European. We know that these are less common inside, but we don't really have a target of where they should be in that distribution. There's been some other great work by folks. Catherine Ross, working at Mulligan's Flat, has looked at early Nancy's and that interaction with bettongs there, and found something kind of similar. You have a real redistribution of that plant at Mulligan's Flat, where the large ones really survive in rock crevices, and the smaller tubers underneath are less common across the open landscape. So again, it's that when we think about managing these things, it's thinking about, we have to manage refuges for our mammals. It could be in the natural landscape, some of the plants need refuges from their predators as well. So what we need to do is think about how we can manage that predation as well. So I wanted to kind of end by touching on some work that we're doing in the Pilliga. So the Pilliga is a partnership site we have with New South Wales National Parks. I think a few folks have gone on field trips or going to field trips there. And we have this opportunity there to look at what's happening before we put the mammals in. Part of that is we have a bunch of extra time because we have one kind of pesky fox in the Pilliga that we can't catch. It's been years now that we've been trying to catch him. We've named him Rambo, and there's been a huge amount of effort trying to catch this pesky fox, and what it's done, this has allowed us to actually do a lot of pretty cool research looking at what's happening while all the feral herbivores, we've done all of our management inside the fence, we're ready besides the fox. So we've started our Ecohost monitoring inside and outside and particularly looking at the threatened plants that we have there. So we have 3 threatened plants at Pilliga. We have, again, I'm trying to get the names correct. We have the Myriophyllum implicatum. It's a small wetland herb that's critically endangered, and it's really common on the edge of wetlands in that zone that gets wet and dry. And when you talk to Sabrina Carter, is the ecologist up there that's leading this work, and when you talk to her, it's actually incredibly common up there in those areas that we have it. And feral pigs are going to be the biggest threat for that, and goats as well. But really, the feral pigs is what we're trying to manage for that. So again, we only have one wetland inside, so we don't have a ton of power to start looking at this. But certainly, we've been able to get rid of the pigs inside, and we have to manage them outside because we don't have the same level of ability to eradicate. And then we have 2 orchid species. We have the pine donkey orchid and the crowbar greenwood orchid. And again, feral goats are going to be the main threats that we're managing for those. Again, we've had this ability to eradicate those inside, and we're managing them outside. And what we've seen in a couple years that we're doing these targeted surveys, and we're doing these in 2 different ways. We have, like I said, we have these long-term eco-health monitoring plots where we monitor the same things at all these sites, and we have veg plots of these. And we've chosen these long-term veg plots to focus our orchid surveys on, where we have seen them and they're more likely to be. So we have 5 of these inside and outside, so 10 total of those. But we also added some targeted sites for these, because one of the things we realized is plants don't always, especially rare plants, aren't always going to fall where you have your long-term monitoring plots. So we actually, went to the historic records, found out what habitat they preferred, and we went out, looked in that habitat, and found high-density plots of these orchids, and put monitoring sites where there's actually high densities of them now, inside and outside, to see what happens. So we do 2 types of surveys for these. We make sure to survey when they're flowering. We actually have quite a few. Sabrina said we have at least 5 other pterostylis up there, and so we have to make sure. So it is tricky for us. We have to dedicate, time to be able to put this aside whenever we need to. But we've been able to do the surveys a couple years in a row now. And what we've seen is that we do, and again, it's only 2 years of surveys. We don't know, if it's just by chance. The Pilliga is having a great couple of years as well. If any of you haven't seen the photos up there, the wildflowers look absolutely amazing up there right now. And so essentially we see that the pine donkey orchid, we do see quite a difference inside versus outside now. So whether or not that's because of the feral herbivores, we won't be able to say. If we get another year this year, we will have another year this year of this data. So at least we'll have a couple years, without the animals in there. And then what we'll be able to do is continue to monitor as the animals are in there. We haven't seen, for the other orchid, the pterostylis, we actually haven't seen a difference. And again, when you talk to Sabrina, she said these are relatively common up there. So whether or not that's good or bad in terms of no difference, hopefully that means we're doing a good job of managing outside as well, because we're not seeing necessarily good or bad. But this allows us, again, one of the great things about this is because we have these plots identified, we'll start seeing these effects. We're able to be more proactive about that management as well. We know where they are. We'll start to be able to see these effects. And then we can be proactive about managing them from, our responsibility to manage these threat species as well. So it's a really, fortuitous opportunity actually having this fox in here to be able to get some of this data and see what's happening, and then to be able to track what will happen once we put the mammals in there. And particularly as we stage and put different mammals in there. So again, when we do these reintroductions, they get staged over time in terms of which animals go in. So we've got bilbies in a breeding area there now, and we've got brush-tailed bettongs that have just gone in. So we have a 500-hectare smaller area where we are starting to reintroduce the animals, let the populations grow up, and then when the fox is removed, we'll be able to expand out into those other areas. So yeah, that's a short summary of what we're doing inside and outside the fences so far.
If you enjoyed this presentation, then please subscribe to our channel. Other presentations from the conference are available in this playlist, with new ones being added all the time.